Hello, everyone. My name is Gayatri Iyer. I'm a fourth-year PhD student in bioinformatics at the University of Michigan, Ann Arbor, and the title of my talk is Application of Differential Network Enrichment Analysis to Decipher Disease-Related Alterations of Metabolism from High-Throughput Metabolomics Data. We have recently developed a novel bioinformatics tool called DNEA that can build differential partial correlation networks from metabolomics data, perform unsupervised clustering of the network to identify densely connected subnetworks, and finally assess the enrichment of these subnetworks using the network-based gene set analysis or NetGSA algorithm. Importantly, the DNEA application can recover robust networks even with limited number of samples in the data, as well as with highly imbalanced sample group design. Figure 1 shows the overall workflow of the DNEA application. Here, the top module shows the options a user has depending on their data design, and the bottom panel shows the heart of the DNEA algorithm that has been previously published. To demonstrate these various functionalities of the DNEA application, we analyzed 3 metabolomics datasets. The first dataset contains measurements of 163 plasma metabolite levels in type 1 diabetic and non-diabetic mice. Differential partial correlation networks from DNEA showed a substantial reduction in partial correlations among metabolites associated with type 1 diabetes progression, as shown here in figure 2A. Of the 9 significantly enriched subnetworks listed here in figure 2B, S1 was characterized by higher correlations in the diabetic mice and was composed of metabolites associated with oxidative stress and changes in redox potential accompanying diabetes. The levels of most of these metabolites is related to changes in levels of glutathione, a potent antioxidant that was not measured in this study. Notably, these metabolites are not significantly differential and would otherwise not be prioritized by traditional analysis pipelines. Metabolites in subnetworks S2 through S6 were more correlated in non-diabetic mice and largely contained sugars and sugar-related metabolites, amino acids, nucleic acids, and nucleotides. Several of these metabolites have been previously implicated in type 1 diabetes as well as insulin resistance. Next, we analyzed metabolomics data from plasma samples of 774 subjects in the Framingham Heart Study offspring cohort, 100 of whom developed type 2 diabetes over the span of 14 years. This study now has a highly imbalanced sample group design. We implemented a modified partial correlation network estimation methodology that substantially downsamples the larger groups, thereby reducing bias towards the larger group and recovering stable and robust edges in the partial correlation network. Now, like type 1 diabetes, we do see reduced correlations among the metabolites with progression to type 2 diabetes. Of the 6 significantly enriched subnetworks shown here in figure 3B, S1 comprised of several intermediates from tryptophan and purine metabolism, as well as TCA cycle. Additionally, amino adipate, a known marker of type 2 diabetes, is also present in S1. Most of these metabolites are slightly elevated in type 2 diabetes. Subnetworks S2 and S4 are composed of di and triglycerides, almost all of which are elevated in type 2 diabetes, as we would expect. Interestingly, S6 shows an interesting interplay between primary and lipid metabolism. This subnetwork is very well connected with many differential edges, suggesting significant changes in the relationships between lipids and primary metabolites with progression to type 2 diabetes, thus generating interesting and testable hypotheses. Finally, we analyzed the lipidomics dataset from the Michigan mother-infant pairs cohort. In this study, the plasma lipidome of 106 pregnant women was measured at the first trimester, M1, and at the time of delivery, M3. Samples were also collected from the cord blood of the infant at the time of delivery. The study measured levels of approximately 700 lipids. Given the large number of lipids relative to the sample size that can result in very sparse partial correlation networks, we implemented a feature aggregation module within the DNA application that collapses highly correlated and chemically similar lipids into singular features prior to network estimation. This step is detailed in the DNA workflow in Figure 1 earlier. This step allows us to make the feature space comparable to the sample space and therefore recover more robust partial correlation networks. We generated DNA subnetworks from pairwise comparisons between the time point and tested the association of these subnetworks with infant birth weight using a methodology called group lasso regression. Group lasso regression prioritized subnetworks containing lyso-PCs and lyso-PEs, as well as another subnetwork containing PCs and TGs to be strongly associated with infant birth weight. Here in Figure 4, we see the top two subnetworks associated with infant birth weight from the M1 cord blood comparison. Here we see the top subnetwork containing lyso-PCs and lyso-PEs, and the second subnetwork containing PCs and TGs. These results are consistent with some of the previous findings that show a strong association of cord blood lyso-PCs with infant birth weight. In conclusion, the DNA application provides a novel methodology to identify data-driven subnetworks from metabolomics data. These enriched subnetworks have meaningful biological significance and can therefore be utilized to advance metabolomics data analysis. Here is my complete poster. Please do check it out. Thank you for listening to my talk. Please do contact me if you have any questions. Here is my email address. Thank you.